Okay, any change in the DNA sequence is mutation. That can be because of different reasons. Number one, due to deletion of some sequence, maybe some nitrogenous base is missing. That can be one case of mutation. Number, okay, do you have any question? Okay. Second thing is by addition of some DNA sequence which changes the sequence basically. For example, if it should be CAG and maybe somewhere T attached, so it becomes CTAG. So there is change in the sequence. It means the properties or the characteristics would change. Okay. Number three is the non-segregation. Non-segregation means when the chromosomes get segregation. When the chromosomes get separated in the mutation, in the mitosis, due to some reason they don't get separate. That is the case in the Down syndromes. So what is Down syndrome? Okay, okay, okay. Maybe due to some reason, okay, in mitosis what happens, the chromosomes replicate or DNA replicate, so there is copy of DNA. Okay, if it's like this, there is copy, so this copy should get separated and these chromosomes should be separated, right? But if in any case due to some reason these do not get separate, it means if it's human, one chromosome is getting 24 pairs and the other is getting 22. So there is, due to non-segregation or due to no separation of chromosome, one cell is getting more chromosomes. Okay, as compared to, okay, we have 23 pairs, right? When they get replicated, they make 46 pairs. So after replication, they need to get divided and each cell should get 23 each. This is mitosis. Okay. But due to some reason, if there is no separation, then one is getting 24 and other is getting 22. Okay. So Down syndromes is one of the case of it. When the chromosomes do not get separate and it results in various abnormal behavior. Okay. In the characteristics. Okay, there is one other which is known as sickle cell anemia. Sickle cell. Now, what happened in sickle cell anemia? Because hemoglobin is a protein. Is hemoglobin a protein? Okay. So, due to this, when there should be like a change, a deletion of something, maybe some sequence is deleted, due to which the protein is incomplete. Okay, some amino acid is missing, let's say some amino acid is missing. This means the hemoglobin would not be able to carry or would not be able to combine enough oxygen. Okay, so this is mutation. Now there can be several causes of mutation. Number one is the ionizing radiations. That can be X-rays, that can be UV rays, anything that can ionize. The radio waves, these can be causes of mutation. Second cause is the tobacco smoke. This one is also in chapter 10, I guess, pathogens and immunity, or 9 or 10. That this is one of the major cause of mutation and can lead to cancer. So, what is cancer? Cancer is basically the uncontrolled growth of cells. So the cells are dividing normally. Due to some mutation, the division or the cell division is uncontrolled. So there would be a mass of cells that are not normal. That is cancer. Okay, when the cells are not normal. So tobacco smoke is also one of the major cause. Okay. Uh, no. Okay. So DNA, we believe that nucleus controls the cell division, right? Okay. So there must be a sequence in DNA that must be controlling the division. If that sequence is disturbed, then this division is not controlled, is not under control. There would be, like, if the cell has to be divided in like 4 times, it might be dividing 8 times. Okay. So it's not about number of chromosomes, it's about the mutation that is causing more cell division as compared to the normal rate. Okay. Okay. uh so we have discussed the sickle cell anemia. Okay, there is one question, repeated question in the past paper, that what is sickle cell anemia and what happens in sickle cell anemia. So you must know the effects of sickle cell anemia. That, okay, anemia is something related to the hemoglobin. Okay, keep this thing in mind. So in this sickle cell anemia, the cells are not globular, or they're not biconcave. Let me give you a picture. They're not biconcave, rather they have a sickle shape. Sickle is a type of tool used in farming. It's like a moon shaped. Okay, so these are not normal cells, these are sickle cells, these ones. Okay, since they are sickle cells, they are not normal cells, so hence the ability to carry oxygen would decrease. As a result, less oxygen is taken in, there would be less aerobic respiration, there would be less production of energy, the person would not have enough energy to carry out the process. It may lead to the shortening of breath. Okay, there are like so many connected problems with this one. So you need to connect what what is the function of the red blood cell and what happen if these red blood cells are not normal. Are you getting my point? Really? I've asked whether you get this point or not, that you have to relate the function of red blood cells with the abnormalities. If there are abnormalities, GGJ. So number one, if there is no oxygen, there would be less aerobic respiration, there would be less production of energy, there would be, the person feel tired or would have fatigue, and what else I told you? The shortening. Exactly. So you have to like relate the things. Okay, in effects. Okay. So I guess we are done with mutation. Now we have to plan the investigation of different concentration of nutrient solution on the growth of lamina. We have to use the same method that they have used. Okay, like counting the number of leaves. What should be our method? Hmm. 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 Okay, so here we have, what is our limiting, like, sorry, independent one is the nutrient concentration, right? Then same volume of nutrient would be okay. Wait. Listen to me. Independent is the nutrient concentration, dependent is the number of leaves, okay, that we count later on. So the constant variables are volume of nutrient solution, starting number of leaves, same temperature, same light intensity, even same carbon dioxide concentration. You can write here like sorry, same carbon dioxide concentration, okay. And then how do we carry out the experiment? After certain number of days, whatever days are, or each day we calculate the number of leaves in different nutrients, and then we can plot a graph, okay. Okay. Yes, yes, yes. Or otherwise you can write would be kept constant. Okay, same number of leaves at the start of the reaction, same volume of nutrient, same temperature, same light intensity, same carbon dioxide concentration. Okay, or otherwise you can write carbon dioxide concentration, temperature, number of leaves at the start, or the volume of nutrient solution should be kept constant. Okay. Okay, figure 3.1. It's about cells. One cell is plasmolized, other, okay, both the other cell is not. Okay, state three items of apparatus that you are going to use to view the cells. Okay, this one is quite small. Microscope. Okay. Okay, so microscope cannot work without light, right? Slide, slide, slide. We can use slide, we can use microscope, and we have to use the light source. For example, lamp. It's a light source. Uh, Without light, you cannot see, because right now we are using light microscope. Make a large image of only these X and Y, or Y only. Okay, X and Y only. Don't make all the cells, make only these two cells. Okay, X and Y. You need to be like very vigilant, or you should have, what shall I say, vigilant eyes. So what are they asking? You have to draw that only. Okay, describe how would you treat cell from a plant epidermis so that they become plasmolized. Why do cells become plasmolized when we place them in a um, it's in chapter number 3. Sorry. Good. Or a concentrated solution of salt or sugar. Then, okay, first word, the very key word is the concentrated solution, and the second one, concentrated solution of what? Of salt or sugar. Okay. Okay, so what you need to do,